All right, so here is the physiological process of drug tolerance. Remember, we go from the VTA cells to the nucleus accumbens cells, and that's what we see here. This cell right here is a VTA cell. Here's the presynaptic terminal. It's binding onto the dendrite of the nucleus accumbens cell. This is the nucleus accumbens cell. Okay, so this would be the medial forebrain bundle, part of the medial forebrain bundle, this axon. Okay, we know that VTA releases dopamine. It's a dopaminergic cell. So during drug use, the VTA cells are stimulated and release dopamine triggering the reward circuit. So here is the dopamine being released and binding to the receptors on the postsynaptic cell. Okay? Now, dopamine binds the receptors and increases what's called a second messenger cascade. It's not a classic inotropic receptor where it simply binds and opens up a channel to let sodium in or potassium out. It's metabotropic. It opens up and it triggers a second messenger cascade. The two second messengers are cyclic AMP and calcium. This is called cyclic AMP and calcium. So a first messenger is a neurotransmitter that would bind. The second messenger usually happens inside the cell. So here cyclic AMP and calcium are activated now, and they're on the move. And where do they go? They go to activate the CREB protein. So cyclic AMP and calcium activate CREB. CREB goes into the nucleus and acts as a transcription factor. Remember transcription factors? Those are things that turn genes on, get genes moving. CREB activates a gene, at least that's what we believe, as the dynorphin gene to make the dynorphin protein. DNA makes proteins, genes make proteins. So the dynorphin gene makes a dynorphin protein. What happens next is the dynorphin protein is released back onto the VTA cells where it inhibits dopamine release in a type of negative feedback, depressing the reward circuit and causing the user to need more of the drug for the same high tolerance.